Hello, everyone. We're here at Cold Spring Harbor Laboratory for the 86th Cold Spring Harbor Symposium on Quantitative Biology. The topic of this year's symposium is genome stability and integrity. My name is Peter Sherwood. I'm a molecular biologist, and I'm joined today by Donna Branzei. Donna, please introduce yourself.
Yes, so I am principal investigator at IFOM, the Foundation Institute of Molecular Oncology in Milan, Italy.
Great, thank you. Donna will be speaking during the symposium later this week, but I'm delighted to have her here today so we can discuss her work ahead of time. Why don't you go ahead and give me a little bit of background information about your research field, and then later we'll go into your current work.
Right. So my lab has a long-standing interest in the DNA repair mechanisms activated by replication lesions and how these events are interconnected with the overall chromosome structure. So regarding the DNA repair events we are studying, we are fascinated by so-called DNA damage tolerance pathways, whose main purpose is to bypass the lesions encountered during replication in order to allow replication for progression and to prevent for collapse with deleterious double-strand break formation. In doing so, however, the DNA damage tolerance pathways can also be extremely error-prone or mutagenic.
Right. So are you going to be telling us about how cells detect certain kinds of DNA damage and how they then channel the repair process into an error-prone or an error-free pathway?
Yeah, we are not so advanced in understanding how specific types of DNA lesions are being channeled into one mode or another, but I will talk about principles that can guide the usage of DNA damage tolerance pathways. What we have learned is that there are two main modes of DNA damage tolerance. One is a mutagenic mode that uses specialized translation synthesis polymerases to bypass the damage and can introduce mutations. The other one is much more accurate. It uses recombination and a switch to the newly synthesized sister chromatids that is devoid of damage to go beyond the DNA lesion and to allow replication for progression.
Great. I understood that you took a genetic approach using yeast to address some of these issues, and you identified some interesting things. Can you tell us about that?
Yes, right. So one of the big questions in the field is how the DNA damage tolerance pathways are being utilized when the choice is being made. And what we have found is that the replisome components orchestrate the DNA damage tolerance pathway that is being employed at replication blocks. And moreover, we found that this process is coupled with parental histone transfer, also mediated by replisome components such as Ctf4 and DPB3/4.
Oh, great. Yes, when I read about Ctf4, which you started with your mutant hunt, it reminded me immediately of a genetic screen that was done long ago in 1989 by a colleague of mine, Phil Heater, who used some of the first yeast artificial chromosomes in a color sectoring assay. So it was wonderful to see that that is still being in use today, that very mutation, or that at least those very genes.
Right. It is in fact a very interesting mutant. It's a chromosome transmission of fidelity 4, right, and it unites DNA metabolism functions with chromosome structure. So at the time when Phil Heater identified the gene, I think he only knew that it is required for chromosome segregation. Now we know that it is a conserved replisome component. We know the structure, we know many of the interactors. We know that it plays roles in DNA damage tolerance in some way, and also in chromatin integrity and also chromosome structure by facilitating cohesion. So it is a very interesting factor. And the initial screen of Phil Heater actually is gloriously moving forward, because Ctf4 seems to be also a very important target in cancers, in over 30 different cancers now.
Right, great. Let's, I'm not sure how much time we have left, but let's talk a little bit about the rest of the study that you did, where you were sort of able to bring together a Ctf4 with another component of the replisome, and tell us about the results of the drug sensitivity and how it affected how cells remember, sense, and respond to DNA damage and what they do to repair it.
Yes. So Ctf4 mutants are exquisitely sensitive to methylmethanesulfonate, which is a DNA damaging agent that introduces methyl lesions into the DNA. And these lesions are known to activate DNA damage tolerance. because CTF4 has many functions, we did not know how they are connected to each other. So we did an unbiased genetic screen to identify suppressors of the sensitivity of CTF4 mutants. And interestingly, we got mutants of DPB3/4, which are non-essential components of polymerase epsilon.
And polymerase epsilon, remind us, is the...
Is a polymerase that replicates the leading strand, so the continuous replication of the leading strand.
Okay. Please continue.
Yes. So DPB3/4 are very interesting because they have histone fold domains, and they can also bind, in fact, to double-stranded DNA and to histones to mediate silencing and to transfer histones on the newly replicated strands. And it turned out that it is this precise function of DPB3/4 that can rescue the sensitivity of CTF4. And interestingly, it doesn't rescue it by restoring a function that was lost in CTF4, which was a recombination defect, the ability of cells to engage in error-free recombination, but rather making the cells much more susceptible to mutagenesis. So the double mutants become drug resistant, and this is due to access of TLS polymerases to lesions and a commensurate increase in mutagenesis.
Right, so it allows more error-prone replication to occur.
Exactly, and this of course could be very relevant also in cancers that accumulate drug resistance upon chemotherapy.
Great. Is there anything else that you would like to add at this point? Or?
Well, yeah, of course, with this discovery...
What are your plans for the future?
Right, yeah. I mean, first of all, due to the findings that it is a parental chaperone function of DPB3, we inquired whether this function is also important on the lagging strand, and we knew very well the factors involved in transferring histones, parental histones on the lagging strand. And it turned out that it is so, and in fact on the lagging strand the defects are even more prominent. And it's very interesting to understand how this collaboration between leading and lagging strand parental histone transfer needs to take place in order to ensure low levels of mutagenesis and to allow cells to engage into the accurate mode of DNA damage bypass. This is one of the things, the collaboration. Then of course, it will be interesting to understand whether there are specific histone modifications on the parental histones that are important for this DNA damage tolerance mode, and where the mutations happen in the genome if some
yes,
regions
you did say earlier when we were talking that there may be regions of the genome that are more prone to certain kinds of mutagenesis and therefore more apt to be repaired in a certain way.
Yes, at the moment we do not know. For this we will need to evolve cells and to see where they accumulate mutations. So to see whether certain regions are somehow protected, either because they contain essential genes or maybe in relationship to the replication timing. We do not know yet, but
great.
It will be fascinating.
Okay. Well, thank you very much for joining me today, and we'll see you later in the symposium.
Okay, thank you very much.